origin, authenticity, and quality of raw ingredients and processed foods are major concerns for the global food market. From sample preparation to test result, food quality control procedures must be safe, fast, and reliable. The sample and assay technology of QIAGEN's mericon portfolio meets these demands. You can extract pure DNA from all of these foodstuffs and more using the mericon sample preparation kits and then analyze that DNA with our extensive range of real-time PCR assay kits. The focus of this video is sample preparation. I'm going to extract pure DNA from this chocolate using the manual protocol of the DNeasy mericon food kit. This DNA will be suitable for identifying and authenticating the ingredients of the chocolate using the appropriate detection kits from the mericon GMO and plant and animal identification range. If I wanted to detect pathogens, I would use the mericon DNA bacteria or bacteria plus kits to extract bacterial DNA, and then the appropriate mericon pathogen detection kits for the assay. Extracting bacterial DNA will be the focus of the next video in the QIAGEN Food Safety series. This is the DNeasy mericon food kit. It uses a simplified, optimized version of the widely used and trusted CTAP method. It's a universal protocol suitable for all food types. Like classical CTAP, it yields pure DNA, but it is also extremely effective at removing inhibitors. The protocol is very efficient, allowing you to process up to 30 samples in as little as 2.5 hours, compared to 1.5 days with traditional CTAP. As I said, I want to extract DNA from this homogenized chocolate sample. Chocolate has high contents of inhibitors like polyphenols, which would interfere with the real-time PCR identification of the DNA. So it's a very challenging foodstuff for food quality control. This sample was homogenized in a knife mill to guarantee that my starting material is representative for the whole food product. We recommend two slightly different protocols for DNA extraction. The choice depends on the degree of processing of the sample. This is the protocol for foods with a relatively low level of processing. The first step is cell lysis. I expose a 2-gram sample of the homogenized chocolate to the food lysis buffer and Proteinase K. Proper mixing is important, so I will briefly vortex the tube and then incubate the mixture for 30 minutes. During this time, the cells release their contents, including the DNA that I want to extract and the inhibitors that I want to remove. After the incubation, I need to cool the sample to room temperature and then centrifuge it. The DNA is in the supernatant, and the bulk of the inhibitors are in the precipitate. The next step is the chloroform extraction with the DNA solution. This removes the remainders of the inhibitors. After this centrifugation, the upper aqueous phase will contain the crude DNA solution. This final set of steps will yield the pure DNA that I can later test via real-time PCR to identify its composition. First, I have to thoroughly mix the aqueous phase for my previous centrifuge step with Buffer PB. Then I pipette this mixture into a QIAquick spin column. I will centrifuge this and discard the flow-through. The DNA stays on the column. To wash the DNA, I add Buffer AW2 and centrifuge twice, discarding the flow-through each time. To release the DNA from the column, I add the final buffer directly onto the QIAquick membrane. After a final centrifugation, I have the pure DNA that I need for real-time PCR. As I mentioned earlier, I could use this to determine the origin or authenticity of the ingredients of the chocolate using a mericon PCR assay kit. So that was a manual DNA extraction from a sample of chocolate using QIAGEN's DNeasy mericon food kit. The same protocol is suitable for a whole range of foods. You can also automate part of the protocol using the QIACube. In the next video in the food safety series, I will demonstrate a fully automated sample preparation, this time focusing on safely extracting bacterial DNA for pathogen detection using the QIAsymphony.